Hello everyone, welcome to my channel. So this is the first video of a series of videos called bioinformatics. I try to tell a little bit of background and then go to the coding part and start doing some coding. And let's start with the question, why we need bioinformatics? Okay, so this slides answer this question very well. So bioinformatics is so what is bioinformatics? Bioinformatics is an intersection of statistics, biology, and computer science. So we need all these three knowledge. So like we need statistics and we need computer science as well as biology. Why we need bioinformatics? So in the past, biologists they were doing just single gene analysis. For example, they had a hypothesis. For example, BRCA1 is important in breast cancer. Then they were gathering some samples, like some tumor samples, some normal samples, and then they were measuring the expression of BRCA1 in these two samples, or they were studying the mutation of BRCA1 in these two samples. But with the advancement of the knowledge and technology, we were able to study thousands of genes at the same time. So for a single gene, a simple like software like Excel is enough, but you can use Excel like SPSS or Prism and do a t-test, a normal statistical test that would do the job very well. But when you're studying all these genes, like thousands of genes or all the genes in the in the genome, like 55,000 genes at the same time, so those methods are no longer capable of answering your question or even analyzing your data. For example, in RNA sequencing data, the FASTQ file that you get as a result of the RNA sequencing data. Like in Windows and in normal softwares, you can't even open that file. So you need something like Linux operating system to like read the file like line by line and do the operation and then like do something on it and then print the output like on the or in another file. So we are going to start our work with transcriptomics, and then we will work our way toward proteomics and metabolomics, single cell analysis, all these things. But we'll start with transcriptomics. And there are several reasons. One of them is that there are a lot of transcriptomic data. There are a lot of RNA expression, microarray RNA-seq data in a database called GEO, which stands for Gene Expression Omnibus. And we can fetch those data very quickly and start working with them. And also, gene expression is very important in cell physiology. Like we know that all multicellular organisms, like humans, they start from a fertilized egg, but then they develop to a very complicated organism with different type of cells, like different type of cells, like brain, muscle, and liver. So these cells, they're very different. They have the same genome, but the phenotype is very, very different. So what makes this difference? It's just the expression, the genes that are expressed. So because of this, it is also very important in understanding the biology of different phenomena like diseases. There are two main technologies for this microarray, which was the first invention in high throughput gene expression analysis, and then RNA sequencing. If I want to give you a short description of the microarray, in microarray we have thousands or millions of probes printed on a solid surface. And then you extract the RNA from your samples, and then you make a cDNA. cDNA means complementary DNA, and we use an enzyme called reverse transcriptase for doing this. It's called reverse transcriptase because it is doing the reverse job of transcription. right? So it's we are making the cDNA, and then we label the cDNA with fluorescent dye. We can have 2-channel or 1-channel, and then we hybridize the cDNA with the probes, and then we wash away the unbound transcripts, and then we image the chip. And the intensity of each spot tells what was the expression of that particular gene. In RNA sequencing, it's the same. So we start with RNA, we do the extraction, we make cDNA, and then so the only difference is we add an adapter to the cDNA, and the adapter does 2 things. First of all, it enables the machine to do to recognize the probe, to recognize the cDNA and start sequencing. And then it also enables you to sequence multiple samples, because you can use probe adapters with different sequence. So you can do actually, you can pool your samples and do the sequencing in one go. And so, and then when you attach the adapter, you increase the number of transcripts, because you must be able to. So this is we call this step the amplification. So you amplify the signal, and then you do the sequencing. So the machine starts to read the sequences. The result of the RNA sequencing technique is a FASTQ file, and it's a long way from FASTQ file to the gene expression matrix, but we just skip those steps. I will make some videos for later on for how to analyze like FASTQ file and RNA sequencing data. So from FASTQ to the expression matrix. But we have to start small. So I have to upload some videos about Linux, and then some theory, and then we will reach to that. But anyway, so the thing that we are going to start, the file that we are going to start our work today is something like this. It's a matrix with the rows with different genes. So you can see like gene 1, gene 2, gene 3 until gene X. It depends. If it is a microarray, it can be like 15,000 genes or 17,000 genes, or if it is RNA-seq, it can go to up to 50 or 60,000 genes. And then you have the samples in the column. So sample 1, 2 until sample X. Okay, we start from here in R. So in order, okay, so I just need to explain something. So R is a programming language. It's a statistical language. It's very powerful. It's very good. And this page that I'm showing you, this is R Studio. R Studio is an IDE, or integrated development environment. So it's just to make working with R easier. But it's not a language. It's not a programming language. So let's search GEO data. Okay, GEO. Okay, so we go to GEO, and the data set is GSE 39132. It's not going. It's wrong. Okay. So okay, let's explore this page first a little bit. So the accession. So I just put the accession number of this data set. And you can see the first thing is status, which says like it in August 2012, this data set become public. The title of it, the gene expression profiling of Hodgkin lymphoma cell lines, experiment type, expression profiling by array. Summary: Hodgkin lymphoma is derived from germinal center, post-germinal center B cells. Overall design: gene expression profiles of Hodgkin lymphoma cell lines were compared to 5 samples of CD77 positive centroblast derived from reactive tonsils. And I call this dataset the Steidel dataset. It's a very nice dataset. I used it in my PhD thesis. It was very useful for me. And it's very nice dataset. It has 10 samples. That's why I chose this dataset for this for first work. And there is one publication associated with this dataset. You can click on this link and go and read the publication. And then the other information that you get, like in this page, when you start saying is the platform, which here is GPL 570, which is an Affymetric chips, yeah. And then it has the samples. So it says 10 samples. The first 5 are centroblast, and then the last 5 are Hodgkin lymphoma or tumor samples. Okay, let's start fetching this data and do some preliminary analysis on that. I'm not going to show you how to install R and R Studio in this videos, but it's very easy. You can search. I'm sure there are thousands of videos on YouTube. Okay, so the first thing that you do each time, you just go to the folder that you're going to work in, Control L, copy the path, then come back and then set WD. So the command set WD, it will set your working directory. And you just copy that, just change this backslashes to slash. Good to go. And directory is a fancy word for folder. Folder, directory, same. So in Linux, we say directory. In Windows, we say folder, okay? So so this is, and with this work, you just tell R that everything is in this directory. If you want to look for something, if you want to save something, so you don't need to type the full path. You can just give it a relative path relative to the place to the working directory. Okay, so set working directory, and then library to load the packages that we need. GEO query and library biobase library Lima or Lima for normalization. And for today, just pheatmap, huh? And library ggplot2. I just always library ggplot2. It's very handy library. dplyr. Okay, so if you don't have these packages, you just install the packages and then the name of the package inside the quotation mark. Or if it is from the bioconductor, you just BioC lights and then colon the name of the package, or simply just search it, install like install GEO query or something, and then go and copy and paste that code, and then it will install it. For the bioconductor, for all the bioconductor packages, it's very easy. There are vignettes for installing the packages and how like they have a data. which you can play around and try the functions that they have. Okay. So the first command that we need is get GEO. Get GEO. But before this command, so what I always do is I will define some variables. These variables, it just makes your code reusable. But before that, because let's Control L, then rm list equals ls. Okay. So I just removed all the variables, all the things that they were inside R. So everything is clear now. So we can start. So, okay. So the series was GSE, insert quotation, GSE, what was it? Just go and copy and paste that one. To C and GPL 570. Okay, so I was not doing very important things. So what I did, so I stored the series accession number and the platform ID in two variables, series and platform. And this is how we assign variables in R. Greater than mark and a hyphen. You can also use equal sign, but this is what I do. So I use equal sign inside the parentheses, and outside the parentheses everywhere else I just use this mark, this sign. Okay. Okay, so get GEO and then series, then say anode GPL true, and GSE matrix true, and dir destination directory goes to dot. Dot means here, current working directory. Two dot means parent directory. And then I met the data folder. I'll okay, so I'm just telling where to store the data in this folder and data subfolder. And I assign it to a variable called gset. Okay, let's run this command. Oh, series not okay. Yeah, sorry. I didn't run these commands. It couldn't find series and platform, and then run the command. I think it found the files. Okay. Now what I need to do is so these gsets is inside a list, and the expression set that we need is the first element of the list. So with two square brackets, we just get that gset. Okay, so if we check the class of gset now, It should be expression set. Yes, it worked. Expression set. Okay. Let's have a look at this expression set. See, it's again a list. It's very complicated. We don't need this. So we want to extract the expression matrix. How we do that? There is a function for that, exprs for expression. Expression gset. And we assign it to a variable called ex for expression. So these are just the name. So you can say whatever you want. Oli, Aleki, I don't know. Whatever you want. But I usually make like meaningful names that I can understand if I come and check my code later. Okay. So we run this command. Okay, so let's check what is the minimum of the exp. The minimum is 1.9. The max. So with these two commands, so you have learned a lot of R commands now. So how to get a minimum of in a data frame, how to get a maximum, how to check the data with head. So you can see it's a nice rectangular data frame or matrix, which the columns are the samples and the rows are Affymetrix probe IDs. When we checked the maximum of this data set, it was 14.7. By looking at this number, I would say this data set, it is normalized and it is in log scale. Because if it was not in the log scale, you would see numbers like big numbers like 200, 2000, 25,000, very weird numbers. But when you see like nice number, not bigger than 15 or 16, it means it is in a logarithmic scale. And let's check. Let's plot a box plot to see if it is normalized. So with the command PDF, we make a PDF. We give it pass here. We put it in results. You can use tab completion. It's very nice. And then we give it a box plot dot PDF. Okay, we created the file. So we just say box plot. That's the command, ex. Box plot expression. Okay, as simple as that. R is very simple. We created a box plot. The only problem is that we didn't. So if you try to open the file now, you would face an error. You just need to turn off the device with dev.off. That's it. Okay, let's go and check inside data folder now. Sorry, no, result folder now. Okay, I think I need to stop the sharing. Share screen again. Okay, see, you get a nice box plot. All of them, they have the same median. So this means that this data is normalized. So they put the normalized data in GEO dataset. But if it was not normalized, it was not a big deal. You could very easily do the normalization inside R. I will show you the command. One normalize. Normalize quantile normalization. Okay, normalize quantile. Okay. So this is the command. Normalize quantile, and then you give your data, like exp. If you have 0, if you check the minimum, no, this is for going to the log scale. So no, nothing. Sorry. So this is one way of normalizing the data. Quantile normalization is one method. There are different methods like FPKM, RPKM, which they are usually used for RNA sequencing data. And quantile normalization is, so the assumption here is that most of the genes they don't have different expression between the samples, so they should have same quantiles, which works very good with the microarray data. So this data is normalized, so I'm not using this command now. That's enough for today. And we just do one more thing today, and then it's just for quality control analysis. So we want to make a correlation heat map. So again, we open a PDF and we give it the pass here, results. No, PDF. Oh, we didn't put it in quotation mark. Here, results, and then core heat map dot PDF, and then core ex, and then dev dot off. Okay. Okay, so we made a correlation heat map. There was an error opening the document. Okay. Okay. Oh, okay. So, okay. So we have this correlation. Sorry. Yes. Let me. Okay. So we have the correlation matrix. And then the command that we need is p heat map core. I need to assign it into a variable. Yes. P heat map x. So, okay, so we run this again. Okay. Let's have a look. Yes. So let me show you. Stop share. Share again. Screen. Okay. So this is the nice correlation heat map that we got. So the only problem is that it is in GSM 95660. So these are these numbers, which they are not, they are no good to us. So what I usually do here is that stop share. I want to share our page. So what I usually do, I define a grouping. So GR equals, okay, so what are the samples? The order of the samples is the order that they were listed in the GEO dataset. So I would say C, and then rep. So I would say norm for normal samples, and then 5 times, because there were 5 normal samples and then 5 tumor samples. Okay, and then rep. 2 more. Two more. 5 times. So you had 10 samples. So this is just the order of the samples. And rep means repeat. So you say repeat this thing like norm 5 times. So let me show you. For example, you say print for printing something, the screen. Print like rep like hello, and then 2 times. It will print 2 hellos. Okay, so that's that one. You can just give it one by one, like norm, 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 and then 2 more, 2 more, 2 more, 2 more, or you can say rep, norm, and then 5 times. Okay, so what I'm going to do is, so in pheatmap we can say row underline label. Or label. Okay, labels underline column GR, and then labels underline row is GR. Okay, so we do the same thing again. Oh, the file is open. So okay, so the file was open in the PDF file. So in order to save the PDF file, you do that, okay? Because of that, he couldn't write the previous file, but I will show you here. So here you can see that, so it labeled the samples too. So the 5 normal samples, they are close to each other. And then the 5 tumor samples, they're also close to each other here. So this is one way to do the quality control of your samples. So if, for example, you see there are 2 normal here, and then two more here, and then 1 normal, and then 2 more, and then normal. So they're mixed together. So it means that your sample, they're not of high quality. So correlation heat map is a quality control method that you can use before going to differential expression. The other method is principal component analysis. But for today, I'm going to stop this video. I will upload next videos related to PCA and how to do differential expression, how to make some plots, and the rest of the works in other videos. I might not be able to upload videos quite often because I'm just writing my thesis at the moment. But just bear with me. I will try to do my best and upload videos at least once a month, let's say. Okay. See you. Bye.